Hello, everybody. Welcome again to another edition of the DCIC Links DCIC Data Science webinar. Today we have an exciting speaker, Bert Goff from University of Pittsburgh, who will be talking about the Microphysiology Systems Database. So without further ado, please, Bert, go ahead.
Thank you very much, and thanks for the opportunity to present to all of you. I'm here with Mark Sherdak, who's the PI on this project, and Luke Bergenthal, who is our chief programmer, who's done most of the development on this project. So if there's technical questions along the way or something like that, there's people here to help answer those. So I want to start by giving a brief introduction to microphysiology systems, because that was really the driver for developing this database. Go over some of the major features of the microphysiology systems database. Have a sort of quick live demonstration of a few features of the database. Talk a bit about the application to a new program by NCATS on developing tissue chip-based disease models for efficacy testing. Talk a little bit about our approach to evaluating reproducibility in these models, and then summarize it all. So the project really arose out of a program from NCATS to develop microphysiology systems for efficacy and safety assessment. And here at the University of Pittsburgh, we were awarded a grant to develop a liver microphysiology system. And in starting to do that, we thought that it would be valuable to have a database that we could use in order to manage our data, as well as to manage reference data that we would use in order to evaluate the performance of our model. But since then, that program actually ended in June this year. But since then, there have been 3 additional programs along the same line to develop microphysiology systems, apply microphysiology systems in different ways, including the newest program, which just started recently, for developing disease models in these microphysiology devices. So the initial program had, I think it was 12, I think there's 12 groups that were developing models, and there are various organ models. There's kidney, liver, neurovascular, female reproductive. It's really a wide range of types of models that were being developed. And the models took on different forms and generate different kinds of data. So on the left, you can see an example of a vascular model where there's a network of vasculature that can be used both to study vascular disease as well as vascularization in cancer models. And it's really very image-based because of the nature of the model. In the middle, we have the human liver microfluidic model that we developed here at the University of Pittsburgh. And it's more of a discrete device that tries to mimic the liver acinus, which is the basic functional unit of the liver. And in that model, we collect a wide variety of data, image data, biochemical data, ELISA data, mass spec data, and then we take all of those data sets, which come from each of the models, and we want to use those data in some way to evaluate the performance of the model with respect to what's known about clinical effects of drugs on the liver. And all of these models have been further developed and in some cases reduced down to arrays of models in microplate format, like you see in the upper right-hand corner, or even one of the newest models that's being proposed uses microbubble arrays, which are really high density arrays of organoids, I guess you would call them, little organ models in a gel matrix. So there's a wide range of types of models and a wide range of data generated in these models. So it's a little bit of a challenge in terms of managing, data for all of those various kinds of models. So why are people interested in microphysiology systems models? I think there's a strong belief that building human microphysiology models will produce models that are more relevant to the human systems. That ultimately these models will reduce the cost of drug discovery and development, reduce the use of animals in drug testing and development, and that these models can be constructed with iPSC-derived cells, human cells, in which case we can build models that are patient-specific and thereby support precision medicine in therapies. And as I said, all of these models are continuing to develop and in many cases are being further miniaturized or built into arrays that allow higher and higher throughput platforms. But ultimately the goal of all of these tissue models is to build models that are predictive of drug safety and efficacy. So some of the challenges associated with this is that none of these models is perfect. They're models. They have, there's certain things that they recapitulate about human physiology pretty well, and then there's certain elements of the models that really are not so consistent with human physiology. And one of the challenges is to determine what is the range of applicability of each of these models. Most of the models still need validation. They all have strong proof of principle data from their initial developers, but they haven't really been tested in terms of their reproducibility and transferability. And in fact, there's one of the NIH or NCATS programs is funding tissue chip testing centers, which are doing precisely that. They're taking these models in-house in a separate laboratory and implementing the models and then testing their reproducibility in their hands. The measurements that are made in these models are, in many cases, more mechanistic than clinical data might be. And so the good news is you get a lot more information about the specific mechanistic effects that a drug has in the organ and in the cells of the organ. But one of the challenges is then how do you relate that back to the clinical adverse events that are observed in the use of those drugs. And we envision that being an exercise in modeling, taking the data that's generated in these experimental models and using that along with reference data from drugs where there has been human exposure, and we do have clinical reference data, and then building computational models that will relate those two. As I said, iPS-derived cells are considered to be high potential in terms of constructing these MPS models, especially with respect to specific patients, but they're still kind of in an early stage of development, and in many cases the iPS-derived cells represent sort of immature cells. For example, in the liver, it's easy to get immature hepatocytes. It's much, much more difficult to get a mature hepatocyte that has the same functional characteristics as a primary hepatocyte. But progress is being made in that area, and we're continuing to see more improvement and expect that eventually iPS cells will be able to provide that source of scalable number of cells with mature characteristics. So a major goal of the microphysiology database is to integrate the data that's been generated in these microphysiology systems models with reference data that comes from biochemical, preclinical, clinical, and post-marketing exposure to these compounds, and use that to establish the clinical relevance of the models, the range of applicability of these models. So this is on the right-hand side, you see a picture of the home page for the Microphysiology Systems Database. It's intended to be a fairly simple, straightforward user interface that's icon-driven. And it's based on a workflow that really goes from study design through implementation and study analysis. And when I say study, so one of the experimental concepts that we sort of built the database around is the idea of a study which would consist of some set of models, of experimental models. It could be as few as a single model or as many as thousands of individual organ models that were treated collectively with a range of compounds or other treatments, but processed kind of in a particular group with a particular goal in mind. So the study is just a collection of these organ model systems. So the database was designed then to create these studies and capture the experimental results from the studies along with the metadata for the experiments. And I think capturing the metadata is perhaps one of the more challenging aspects of the process, because each individual laboratory kind of handles their kind of metadata, in their own way. And what we've done is worked with these tissue chip testing centers in particular to try to reduce that down to a standard format in which they can encode the experimental protocols that are used, so that when we have the data, we can associate the data with the experimental treatments. And so once the data has been imported, we provide tools for the individual data developer or data provider to review and verify and sign off on their data. And then once they do that, the data gets locked in the database, so it can't be changed anymore after that. We provide a means for the data provider to share that data with colleagues, and they can specify who those colleagues are, and then that group of people all have access to that data regardless of where they are in the world. And then there's a means I'll show you in terms of being able to export the data for a variety of reasons, maybe to do some custom analysis, maybe to do some custom graphics, and maybe to combine data sets from different organ models or different studies in order to do some cross-study or cross-analysis. And then we're working on tools to provide a means of establishing concordance between the tissue chip and disease models with the reference clinical and preclinical data. And as I mentioned, there's this new program to develop these disease models. That program just started, and we're currently just starting to work with these disease model developers in order to support those. And we're anticipating from what we've seen of these disease models that there will be even somewhat more variety in those models than we encountered in the first group of models that were developed, principally focused on toxicity assessment. And we're always sort of constantly looking for data sources that will provide relevant reference and clinical data. So this is kind of a conceptual drawing of the interactions and the context of the Microphysiology Database. It's a web-based database. It's administered here and hosted here at the University of Pittsburgh. We've been doing all of the development on the database, although the development of the database is managed in a GitHub repository, so others could participate in that development. There's a series of users. There's the, what's on the right-hand side is referred to as data uploaders. Really, they're more than just uploaders. They're data uploaders, data reviewers. The data that they provide to the database is managed and controlled by those data providers. There's a group that I refer to as study monitors, which consist of people at NCATS. There's a IQ Consortium, which is a consortium of pharma representatives that are participating in this Microphysiology Systems program. And so they're keenly interested in these organ models, and they have signed non-disclosure agreements so that they could get early access to the data that's being generated by these models. Ultimately, the data will be made available to the public, but that's the data available to the public is controlled in a way that the data providers can control when that data is made accessible to the general public. And then there's a series of reference data sources that are used in order to collect reference data that we will use in order to establish concordance with clinical and preclinical results from drug treatments. And there's a variety, so there's two types. We have the ones that are listed on the left there as curated data sources. Those are data sources where a person has to go and extract information from the data sources because it's not really in a convenient format for automatic data extraction. And then there's a number of linked databases which are either dynamically linked and data is retrieved from those on the fly as needed, or in some cases relevant subsets of the data is downloaded and cached locally, and then those caches are updated regularly in order to provide access, more immediate access to the data that's needed in order to analyze particular compounds or organ models that are being used in the database. All of the back-end and front-end components in the database are open source components, and the data sources are all publicly accessible data sources, and so all of that makes the database a very open product itself, and we intend the database to be open and available to all interested parties, and as more data gets released to become publicly available, will be available to anybody and everybody. So the workflow in terms of capturing the metadata and capturing the data into the database involves interaction with the data providers in order to establish for their model the context of the model, what kind of a device is used, what kind of cells are used, how is the model constructed. All of that information gets parsed into the database, and we have some standard formats for the providers to be able to give us that data in a way that we can parse it into the database for reference access. Then each study, for each study there's a study design that defines what the treatments are, how many models are being used, what the timing is of treatments, and all of those important factors that you need to know in order to be able to interpret the results of the data. And so we capture all of that in the database. And then the study data itself from all of the various assays, and in most cases there's a relatively wide range of assays that are run, and the data from all of those assays needs to be associated with the metadata that's relevant to those particular chips and assays, and then stored in the database. And so we manage all of that. And then in the end, some description of the study, what its goal was, or the purpose of the study, in order to give kind of a high-level view to the user who's opening up to look at this study of what is the study about. So that also helps in terms of the interpretation of the data. So then from this data we can do analysis, statistical analysis, generate reports, and do computational modeling, as I mentioned earlier. So one of the I guess tools that we've developed and provide to the people who are generating data to go into the database is what we call the data import tool. And basically it's an Excel spreadsheet that can be used to format the data from a variety of input starting formats into a format that's compatible for direct upload into the database. So by using the data import tool, users can pretty easily take data that's perhaps data that came from a plate reader or some high content screening system, and just paste that data into the data import tool, have it be formatted in a tabular format that's compatible for upload, and then the users can either upload it themselves or they can send it to us and we can upload it. And we do it both ways depending on the user's preference. So here's where I'm going to try to go into the database, and hopefully I can do this smoothly, and you can see a little bit of what some of these things look like. So I'm going to open up some screens here. So hopefully this is coming through clearly. So this is what the metadata that goes with a study looks like. And so there's, as I mentioned, there's a description of the study. There's some information about the organ model that's used in the study, the protocol that's used, that was developed for the study, all of the assays that are in the study, and what methods are used in order to read out those assays, and then some places to add some additional data if there's data that perhaps doesn't fit into any of those other categories. A file can be uploaded and used as a reference for this particular study. So each study typically is focused on a particular model, although the way the studies are defined, there's no reason why a study couldn't consist of many different models. But at this point in time, most of the studies are related to a particular model. And in the database, we have model descriptions, which this is an example of our liver microphysiology system, the model descriptions. And so the model descriptions are fairly brief. They tell sort of whose model this is and what it's called, but also have places to upload some images of the device, such as on the bottom. We use this Nordis microfluidic device for the model, and the actual liver model is this, as you can see my cursor, is this small region in here. But this is, there above it, there's a blow-up of a cross-section pictorial view of what that model looks like. So it's a layered model with multiple cell types layered on top of each other in a flow-through microfluidic device. And so each of the models can have pictures and other information about the model that's used, that can be used as a reference for understanding what the model is about. And each of the devices, there's also, I'm not going to show it, but there's also a separate table that stores all the physical characteristics of the device. So we have in the database a lot of information about compounds. Again, from the idea that understanding what the compounds do biologically is important in terms of interpreting how the compounds interact with these biological models. So one example is, and I mostly brought this up as an example of a query interface. So as you can see here, so this is a query interface to generate a table of bioactivities that would be associated with a selected group of compounds, for example, or selected compounds that are known to have activity against a particular set of bioactivities. So you can choose here different bioactivities, targets associated with those bioactivities and compounds. You can choose single compounds or large groups of compounds, and you can decide whether you want to see bioactivities that are associated with cells or organisms, single protein targets, for example, from humans, from rats, from dogs or other organisms, and then generate a table of those bioactivities. And so you get a table of the compounds and then the compound targets, the organism involved, and then what the bioactivity is. So that's a convenient tool for exploring compounds that you might want to use in a study, for example. There's also some just normal chemical information, physical chemical information about the compounds themselves. So this is just a list of all the compounds. There's quite a number of compounds. Most all of these compounds were selected because they're somehow related to the organelles that are being analyzed in the database at this point in time. But each of those has links back to ChemDL, so you can go and get even more information about the compound if you want, drawing of the structure, compound name, that sort of thing. All those kinds of things that typically you would want to see in terms of having some information about the compounds. So each of the studies, as I mentioned, is composed of a number of chips. And here's an example of a chip setup. So one of these chip setups would be a component of the study, and it tells what device it is, what model it is, what assays, what compounds are going to be used at what concentrations and with what timing, and then also what cells are used in order to construct that model. And so in the database we store individual cell types as well as lots of cells so that characteristics can be compared across a particular lot of cells, for example. So when a study is run, then we end up with all of these setups these are all the chips that were used in that study, and you can see there's a variety of compounds that were used on those chips with different compound concentrations and different timing of exposure of those compounds. And each of the chips generates a series of readouts. That's a set of measurements associated with that particular chip. And so for a chip, there's some information that reiterates the assays that were run on that chip, and then the assay results for that particular chip, and a data table that allows the data to be annotated with cautions, exclusions, notes, and other kinds of information that may be important in terms of understanding an outlier or even just the response in a particular measurement. So while you could go and look at each chip independently, we have a study summary that summarizes for that study all of the chips. So this is an example of, for each assay that was run in that study on each chip. So you see here it's a list of all of the chips that were involved in that assay, in that study. And then these are the results at different time points for this albumin assay in terms of nanograms per day per 10 to the 6th cells. So it's a normalized measure of albumin secretion. We also have bioefflux, which is an image-based measurement. Testosterone clearance, which is a mass spec-based measurement. Lactate dehydrogenase, which is a biochemical assay. TNF-alpha, which is an ELISA-based assay. So there's a wide range of assay types. You can see that the sampling for LDH was much more frequent than it was for the other assays. All of these graphs are generated automatically. They're displayed here by device, but you can also click and convert it to a display by compound. So now you can see that each of these bars represents a particular compound in that device at a particular time point. So you can compare across compounds. And then if you want, you can convert all of these to percent control to make it even easier to compare the response to each compound over time, or or compare the results between different compounds. So there's a good degree of flexibility. It's not a super flexible graphing package or anything like that, but to address that need for, in some cases, maybe some more sophisticated graphics, we have the ability to download the data. So you can click here and you get a data table that in Excel that you can then take to any other application of your choice and use it to generate whatever kinds of plots you would like to make. So there's a good degree of flexibility there. We're certainly in the process of enhancing our graphics in the database, but in the meantime, we do have a way for users to be able to generate other kinds of graphics and other kinds of analyses. And then I want to show you one of the analytical tools that we have in here, and this is a link to the FDA adverse events database that we use in order to evaluate for particular compounds and compare across compounds the adverse event frequencies that are associated with those particular compounds. So here, for example, we can compare compounds, so we can pick compounds, and I'm going to pick these two particular compounds because I know a little bit about them, entacapone and tolcapone. Tolcapone was originally used as a Parkinson's disease treatment, but was found to have a high frequency of liver adverse events and was given a black box warning. And entacapone came out later as a safer alternative for the same treatment. If we want to look at their effects on liver, we can search just on liver, and so we see that we have the liver adverse events, and we can pick some of those here and look at some plots below. So these are normalized adverse event frequencies that are normalized to a CDC database that we also link to in order to be able to assess the frequency of adverse events relative to the frequency of drug usage. And so you can see here that tolcapone has a much higher frequency of adverse events relative to the frequency of usage. The frequency of usage is much lower. If you look up here, you can see that tolcapone, there's only an estimated usage of 10,000, whereas there's more than 10 times as much usage for entacapone. And if we go here and we look at the raw counts, which are now in these light-colored bars, the raw adverse events counts aren't that different. They're still a little bit higher for tolcapone than entacapone, but until you normalize them, you might be sort of misled into thinking that tolcapone and entacapone had similar adverse event frequencies, but in fact, entacapone is much lower. So that was kind of a novel approach to using the adverse events data that we integrated into the database in order to be able to better interpret compound effects on organs. So this is just a little more enhanced version of what I showed you on the previous slide, or previous from the database. And this just shows you that if you look, you can look across multiple organs. And it's interesting to note that the adverse event frequency in the liver is very different for tolcapone and entacapone, but in other organs, not so different. So one of the newer things that we're doing here in terms of development on the MPS database is working to support all of these new disease models. And as I mentioned, this program just got started. There was a kickoff meeting earlier this month. So we're just starting to get familiar with all of these various models, but one of our major goals is to support all of these models in the microphysiology database. And the concept is really to kind of develop portals that would be relevant to each model, in which case, you know, a portal would provide some reference information about the disease, about experimental data from normal and diseased states, and have that sort of in conjunction with the data that's generated in the actual experimental model in order to be able to easily make comparisons between clinical data and the organ model data. And so we'll be, you know, adding curated preclinical and clinical data associated with each of these organ models, and then continuing to develop tools in order to be able to associate those data with the models. We're also working on developing measures of reproducibility for all of these various data types that are generated in the database. You know, in particular, a lot of the data is of the nature of being a time course, and so we're working on tools so that we can look at chip-to-chip, day-to-day, and even site-to-site variability in the data that's being generated. So we've evaluated a number of different metrics for comparing reproducibility. We really like the intraclass correlation coefficient, and I'll show you a little bit of what that looks like. But we're also looking at standard statistical measurements of those differences. So this is the intraclass correlation coefficient. It's used actually pretty frequently in terms of medical devices and characterizing the reproducibility of measurements from medical devices. We've established this scale here to sort of give a classification of reproducibility based on a numeric value of the intraclass correlation coefficient. So these are just a couple of examples to give you a feel for what that looks like. So here's a time course from a series of chips where the intraclass correlation is quite high, the reproducibility looks quite high, so it gets classified as excellent based on the correlation value. This correlation value is a bit lower. It falls down into the next category of moderate. But you can see that the reproducibility amongst all of these chips is pretty good. And then an example of poor reproducibility, in this case, different chips seem to be producing responses at different times. And the intraclass correlation coefficient is low. It's actually negative in this particular case. But each of the chips also gets an intraclass correlation coefficient, so you can also look at the individual chips and identify the ones that are kind of the worst case in terms of being consistent with each of the other individual chips. And so there's a lot of information there that helps to filter through the data and make sure that the data that is generated that we understand sort of where the inconsistencies are, and we can address that in some way. So, in summary, we believe that MPS systems, they have great potential to simplify, reduce the cost, and personalize the development of new therapies, but there are some development effort that needs to go into that, in particular in terms of establishing the reproducibility and the concordance of these tissue models with clinical data. So we're currently supporting these two programs from NCATS, the Tissue Chip Testing Centers program, and this new disease modeling and efficacy testing program. And a major goal for this project is really to be able to aggregate data from all of these various models, accommodating all of the differences in terms of their construction and data types, and to provide the user with the tools that are needed in order to evaluate the capability, the reproducibility, and establish the clinical relevance of the models. So I thought I'd end, actually I have acknowledgements at the end, but I thought I'd end here with some, in looking at the links website and thinking about, what would be what kind of interaction with links, might be useful. And it seems to me that you have a lot of well-documented standards in terms of metadata formats and content and all of that, which I think could be really useful to us in terms of ensuring that the metadata that we're collecting is complete, but also is consistent with a standard, like the standards that I saw on the website. I think there's potentially an opportunity to interact with other links data providers. Just in glancing on the web page, there were several of the apps that looked like they were potentially useful to us in terms of having reference data and reference information about diseases, disease models, pathways associated with those diseases, cell lines that might be used in particular models, and even repurposing drugs. And then I think, there's an opportunity that some of the data that gets collected in the Microphysiology Systems Database could be a data source for other activities in the LINKS program. So these are the collaborators that have been involved on the UPIT side in terms of developing and implementing the microphysiology database, and Texas A&M and MIT are the two sites that are the tissue chip testing centers, and we've been working with them. They've been great in terms of providing us with input and ensuring that the tools we develop were useful and simplified rather than complicated their efforts in terms of generating data and transferring it into the database. So I'd be happy to answer any questions, or other people here can answer questions if there's questions in the group.
Thank you, Bert. That was great. So please, anybody, questions? So maybe I can ask you a question. So I mean, the information that is stored is pretty interesting to me, at least. It seems that you processed or at least exposed Campbell bioactivity database, for example, among other things there. So it seems to be like this integrative aspect seems pretty strong, and I'm curious though, how do you didn't show any programmatic, right, access to the databases and information there. Is that something that is on your plate? And how standardized things are that would say allow for maybe somewhat automated access and reanalysis of the data for different assays?
So yeah, that's a great question, and I think at this point in time we haven't implemented any kind of direct access to the database. I think certainly we're interested in doing that. Most of our focus has really been so far on getting data and getting it into the database and providing user interactivity in the database. But certainly I think in the longer term, we're interested in that. It's also, I think, in terms of priorities for us, much of the data that's been going into the database is still sort of being kept private to some extent within the Tissue Chip Testing Center's program. I think that NCATS is certainly has expressed to us that eventually all of this data should be made publicly available, and eventually probably means sometime on the order of a year, so maybe a year from now. And I think that's kind of a good time frame from my perspective to think about once that data is publicly available, that it would be useful to provide some kind of a interface to be able to directly access that. So I think the answer is, yes, eventually we intend to do that. It's just, I think right now, putting the cart a little bit ahead of the horse.
Okay.
So if there's no more questions for me, can I ask a question?
Please, go ahead.
So I know there's some centers from just looking at the LINCS website, and I didn't really know much about LINCS until Lucy Lowe at NCATS suggested that there might be an opportunity to interact with LINCS in some way. And so I went and looked at the website, and it looks like there's some centers that are funded under the LINCS program. But then there's also all of these apps that seem to represent, in many cases, data sources or other kinds of databases, that all of which are centered around, I guess I would loosely put it at evaluating cellular data with respect to targets and pathways and networks and all of that. But it wasn't so clear to me that collection of apps. Are all of those apps originating out of the centers that are funded under the LINCS program, or is there some other some other process by which these connections to these other data sources and apps has been created?
Well, so there are a couple of sources actually within the LINCS itself for these apps. So one is data generation centers and applications that they developed to access and analyze data they generate. The other, maybe a bigger set of apps, has been generated by Data Coordination and Integration Center, and that's us, right? We are three groups: Cincinnati, Miami, and Mount Sinai, and we have created quite a repertoire of the apps themselves, and some of them do interact directly with LINCS data, but majority of them at least in some way integrate other data resources or information resources. So So the DCIC objective is to, I guess, provide for integration of links data that is generated by different data generation centers, but also to enrich this, right, and integrate also other data that can help in the or synergize with links data to provide a more meaningful insight into these perturbation signatures. So that's maybe where this is coming from, and then where the apps are coming, and then also we are some of our apps actually leverage other web services and or applications that have been generated by wider community, but they are not necessarily featured on our website. So I suspect that all the apps that you have seen on Link's website have been generated by either data generation centers or data coordination and integration centers.
Okay. That makes sense. I mean, you know, it sounds like that's sort of a very similar concept to ours in terms of, you know, wanting to reach out and identify other data sources that will enrich the data that we're collecting from these organ model systems.
Right, right. And so, you know, when I look at your database, I immediately see a resource that I think would be a great addition to this landscape, right? Data landscape that we are creating for integrating with links data. But I guess the question of this data being still open and then appropriate access to it would still have to be resolved. In terms of links, right, links is actually the main focus is generation and sharing of data, right? So all the links data is publicly available pretty much upon generation, and one of our objectives is to basically push it out to the community as fast as possible and in as many different ways as possible, so that it can be used by the community. So it's slightly different, I think, focus probably in this sense.
Yeah, I think that's true. I mean, I think also, so I didn't really notice that any of the apps were focused on sort of clinically relevant data sources. Is that true? That, I mean, you know, I know that you're kind of focused on cellular perturbations and you know, cellular responses and pathways and networks and all of those things, gene expression and all of that. But is there has there been any consideration given to also trying to link in what might be relevant sort of disease data or clinical data? Or is that just considered to be outside the scope of the LINKS program?
I don't think it's considered to be outside of the scope. It is in terms of data generation, right? LINKS generates data from these relatively high throughput models, right? So although there is an effort, right? We also have iPSC models. There are a couple of centers that are focused on iPSC models, and they are also looking into some of the tissue models that would probably be captured or covered by your project. So there is a range of interest in terms of data generation. Again, there is some iPSC like differentiated neurons and the cardiomyocytes as being used as the model, which would, I guess, push it closer to something that could be considered directly relevant, clinically relevant. But also the whole concept of links is to I mean, maybe not the whole concept, but the underlying the motivation maybe for it was this concept of connectivity map, right, of mapping disease signatures to drug signatures. So diseases are embedded in our objectives. Is just not necessarily, we don't generate clinical data, or the models are a bit more towards a high throughput versus this high content model. So in terms of apps, right, there are apps that are mapping or trying to establish connections, say, between perturbation signatures and disease signatures and that sort of things. But it's a way, again, of trying to make links data relevant in the context of diseases research, but we don't necessarily have clinical data.
Bert, this is Damir Sudar, and I'm associated with the OHSU Data Generation Center. One thing that seemed pretty obvious from your talk is that quite a few of the devices generate imaging type of data. Do you, in the database, have the ability to have the image data, the underlying image data, available to be able to drill down to from the visualizations?
That's actually something that we're just working on now. We certainly had that intent from the start, but you know, priorities are priorities. But we now have a complete study with a complete set of images that were generated along with that study, and we're working on integrating that in such a way that people will be able to look at particular data points and then bring up images as a reference for interpreting those data points, and also, if they wanted to do some additional analysis on the images, to be able to access those images.
Okay, yeah, that's interesting. We've been working on giving access to the images that we create within our center through a cloud-based resource where we host those images so that people can access them without kind of putting a burden on our resources, our computers, our storage systems. And the idea is then that eventually they can pay for access and running on the cloud resource themselves when they want to.
Yeah, that's a good idea. I mean, so far, the amount of image data that we have isn't so large that we have to worry about the storage just yet. But as it grows and we get more, that sounds like a really interesting option.
Okay, I'll be in touch on that.
Okay.
And just maybe to mention this one more time. So if you think that certain portions of links data that could enrich, right, your data sets and would be something to integrate with your information, let us know, and DCIC can assist you certainly with using links data in this sense, because that's one of our objectives. So.
Okay, that sounds great.
All right, well, thank you again for the great presentation, and I hope I'll see you all later at the next webinar. See you all later. Bye.
Okay, bye.